Welcome to this session on deuterated standards for LC-MS analysis, what they are and why they matter. In analytical chemistry, especially in LC-MS workflows, accuracy isn't just a goal, it's a necessity. Today, we'll explore how simply replacing a few hydrogen atoms with deuterium can dramatically improve the reliability and confidence of your results. Whenever we perform LC-MS analysis, we face real-world challenges. Instruments drift over time. Ionization efficiency fluctuates from sample to sample. And during sample preparation, some amount of analyte can be lost. These issues can significantly change the final signal intensity, sometimes doubling or halving results if left uncorrected. That's why we use internal standards. They help us correct for variation, adjust for loss, and maintain data accuracy across runs and experiments. A common approach is to use a chemically similar compound as an internal standard. But similar doesn't guarantee identical behavior. Even small structural differences can cause changes in extraction efficiency, chromatographic retention, and ionization response, which reduces correction accuracy. On the other hand, deuterated standards are almost identical to the analyte, just slightly heavier, making them the gold standard for accurate correction. So what exactly is deuterium? Hydrogen has one proton, deuterium has one proton and one neutron. That's the only difference. Chemically, the two behave nearly identically, but because deuterium adds just a bit of mass, a mass spectrometer can easily distinguish the analyte from the deuterated standard. This same molecule, slightly heavier concept, allows precise tracking and accurate quantitation. When a deuterated standard is introduced into the sample, it co-elutes with the analyte, meaning both travel through the chromatography column at the same time. They also ionize with the same efficiency and experience identical extraction and preparation losses. Because any change affects them equally, the ratio between them remains consistent, and that ratio is what produces highly accurate results. Let's take an example. Measuring caffeine levels in plasma. Instead of choosing a related compound as the internal standard, we use caffeine D3. Both caffeine and caffeine D3 co-elute and ionize alike, but show up at slightly different mass values, 195 and 198. By calculating the ratio of their signals, we eliminate instrument and preparation variability, resulting in precise quantification. Using deuterated standards translates to clear benefits. You get higher quantitation accuracy, better reproducibility between experiments and laboratories, and smoother method validation. Most importantly, it strengthens confidence in regulatory submissions and ensures your data can withstand scientific scrutiny. In many regulated industries, these benefits are not optional. They're essential. However, deuterated standards must be chosen and handled carefully. In some molecules, deuterium can exchange back to hydrogen, especially near reactive groups like hydroxyl or amines. So it's important to verify stability, confirm the number of deuterium atoms, and ensure the standard is certified and properly stored. Good lab practice protects the integrity of the correction. Yes, deuterated standards can be more expensive, but the true cost of not using them is far greater. Failed product release, invalid clinical or research data, or regulatory setbacks. In short, the cost of inaccuracy is always higher than the cost of doing the analysis correctly. Deuterated standards play a quiet but crucial role in ensuring accuracy in LC-MS analysis. They allow us to confidently say that the numbers we report reflect reality, not instrument variability. And in science, accuracy isn't just helpful, it's foundational. Thank you for watching.